Good afternoon, class. I welcome you back again to Metric Hub, where today we are going to continue with our lesson 2, chemical pathology. I'm sure that our first presentation and our first lecture, you did understood and you saw the validity of our time. Today we are going to continue talking about the whole blood sample and other types of samples that we are supposed to look at. So without wasting time, let's just look on the notes that I've displayed on the board there. Whole blood sample. It is viscous liquid, dark red in absence of oxygen and bright red when oxygen is present. So whole blood sample, we are saying it is viscous liquid, that is dark red, and in absence of oxygen. When oxygen is there, it's not there, it is dark red, and it is bright red when oxygen is present. And then storage requirements, it should not freeze as this leases the red blood cells, and temperature should be between 4 to 8 degrees Celsius. This is just the storage facilities for whole blood sample. And then all samples must be treated as infectious biohazard, as to say that they can be possibly causing, or they can cause possible diseases. And then blood is normally sterile, but may not, but may contain infectious agents such as HIV, hepatitis B, and syphilis. We are looking at the possibilities of diseases that might arise in terms of infections as a result of blood. So I'm sure that if you can remember, or if you can check very well, having in contact with open sores or open cuts with blood that has got HIV, a person can contaminate the disease, or else also hepatitis C, as well as syphilis. These are contagious diseases, or diseases that can be passed on as a result of having in contact with the blood that has got such infections, or have got such viruses and stuff. So what we are saying is that blood is, of course, sterile, as to say that it might not allow bacteria to live there, but however, we are saying it can cause various diseases such as HIV, hepatitis C, and syphilis. And then number two, let's look at the plasma. It's very watery. The plasma is the watery part of blood. When red blood cells are spun out, you see the blood plasma. I'm sure that if you can remember very well from your previous lessons and lower level classes, blood plasma, it is actually that watery substance that comes out after a wound is bleeded and the bleeding is stopped. So what we are saying is that it is the watery part of blood when the red blood cells are spun out, and it is light straw yellow in color, usually clear, but may be tubid or milk after fat meals, and it will contains since it contains fibrin. So what happens is that the blood plasma also, we are saying that we need to understand that it actually is the watery part of blood, which we are saying found out when the red blood cells is spun out, and it is white. It is actually light straw in color, which is also clear, and then can also be milk after fat meal. If a person eats a fat meal, the blood plasma can also be what? It can also be milky, and it will contain, since it contains the fibrin, and then it usually freezes also at 4 to 8 degrees Celsius. Urine, we are talking about chemical pathology class. Don't forget it. Urine, it is the midstream samples which are usually clear. We say that the midstream is the urine that is collected after a first sample or a first amount of urine has been released, and then in between the urination process, the midstream urine is collected. Then we call that midstream sample or midstream urine, and it's usually clear, and then watery non-viscous liquid. Color might vary, but is usually pale or dark yellow in color. I mean, if you can remember that from the previous lessons, we also identified that the midstream urine is collected whenever we are talking about pathological examination, because it is the one that is clear and then it gives the reliable results that we would want. So total urine samples can be cloudy and also can be tepid, indicating presence of cells. Urine, it can also contain some dissolved salts and some dissolved minerals. So hence it can also be of that color. And then handling midstream urine is usually or is relatively free from microbes. We are saying, I mean, it is known that it doesn't contain any certain disease-causing organisms, and then it can also be used for drug analysis, and legislative protocols may be required in regard to the handling of it. So it does not contain any, I mean, it is free from microbes, and also it is suitable, it is quite suitable for drug analysis again. Uh, Feces feces are highly variable in appearance, but generally the stool should be firm unless diarrhea is present. A normal healthy person should just have a solid stool, which we are saying it should be just showing that the person is healthy and there is no diarrhea. Not unless it is diarrhea, that's when there is, or it can also not be firm. And then color is dependent on diet. When we are looking at feces examination for pathological examination, we are saying it also depends on the diet that the patient has at diseases and disease processes, but it ranges from black to brown or yellow to white, and there's a bad smell, of course, as to say that if there is the type of I mean the type of diet or a meal a person has ate also affecting the smell as well as the color of the stool. I'm sure that we can also agree. Handling feces may contain a lot of bacteria and are significant sources of contamination in the laboratory. That is one key thing that you can also understand, that you need also to understand that handling of feces can also lead to bacterial infection because it contains a lot of a lot of bacteria, and then it is got also, I'm saying it is prone to multiple contaminations in laboratories, and thus a lot of care is required when weighing or homogenizing the feces. That is one other thing that as N5 learners we must also take note of. And then cerebrospinal fluid, like I said before, that it is the fluid that is used or that is contained in ventricles, and it is also mostly contained in the subarachnoid space, which it is also important for lubrications of joints and other body reflexes could also or are taking place or can occur as a result of the CFS, the cerebrospinal fluid. So it's clear, it is usually clear liquid and may contain blood as a result of the collection process. As you can see, we said it is collected through the lumbar puncture, which also we said again that which I had also indicated to the class that uh, it requires specialized equipment, which we said the third and vertebral column is where it can be collected, and then it may be cloud if it contains bacteria. It is, however, normally sterile, as to say that it might not have or it might not allow bacteria to survive within it. Hence, we are saying it is normally sterile. So that is about the cerebrospinal fluid. And then still on that, we also need to look at the mucus or sputum test. The mucus or sputum test, chemical pathology. Don't forget that we are still looking at samples that can be used for chemical analysis. Mucus or sputum, viscous fluid or suspension, usually clear to milk. Uh, Infection may normally be seen by yellow or green coloring, that is indicating infection on the mucus or sputum, which is to say that when it is yellow or green, that it can also indicate that there is infection or the person is not fine. And then if the person is fine, it is actually usually clear to milk color. I mean, that's the color. And then handling, in terms of handling, it must treat mucus or sputum from a tubercular patient, the tubercular patient, with special care to avoid also aerosols, which we are saying also these can also be other respiratory infections that might arise as a result of the handling of sputum or mucus. So patients who have got TBs and stuff, they must be treated and handled with care to avoid also such kind of contaminations. And then we have got aspirates, which we are saying from lungs are usually clear to lung, to milky. Gastric secretions are usually yellow to greenish brown with the possibility of suspended materials. Uh, with the possibility of suspended materials that can also be found there. It has to be properly handled to avoid also spills, to avoid also spillages in terms of the handling of what? Of aspirates. I'm sure that you can also need to take note of that. And then sweat can also be used for chemical pathology class, which is that which you need to understand that it is clear to milky, but usually absorbed on paper or swab. It usually have very small samples, but that needs to be carefully handled also. Sweat, we can, in most instances, it is quite understood that the samples of sweat are quite very small, and therefore they must be carefully handled. Again, again, semen, I'm saying these are secretions that are released from reproductive organs, if you can remember quite well, which we say it can contain the ejaculated sperm cells, and also that fluids that enable the sperm cell to move. So semen is clear to viscous liquid, may be kept warm prior to immediate analysis, and long-term storage will require special diluents and storage at about less minus 196 degrees Celsius, especially liquid nitrogen. So what we are saying is that semen can also be analyzed for chemical pathology, where the pathologist also must make sure that it is kept warm. It must be kept warm towards or with immediate analysis. And then long-term storage also requires some diluent and special storage, which is at minus 196 degrees Celsius, which has got to be done with liquid nitrogen. That is about the semen. That is on semen. And then tissue slices, biopsy, and all organs. I once mentioned before in the previous lesson again that there is what is called biopsy and also other organs that might also be taken for chemical pathology. They are of various appearances and usually small, removed during surgery. According to, they must be removed very small, because we say that these are tissue slices and these are samples, which we say that again, they are a representation of the uh, whole organ from which they are taken from. In terms of handling, samples of biopsies and tissue slices are very small, very easy to contaminate, and usually sent for testing in sterile saline to prevent drying out. So that is one other condition that you need to understand, that they are very easy to get contaminated. They can be contaminated very easily and usually sent for testing in sterile saline to prevent drying out. And then these, we must understand that they must not dry out. Drying out would also cause biased results. It might also cause biased results. So please, class, note that the biopsies and tissue slices must always be moist, must be fresh, and also they must be handled with care, because they are very small and very easy to contaminate as well. Reasons for collection of different biomedical samples. One, we are saying blood to detect the reasons for collection of different biomedical samples. One, we are saying blood to detect bacterial composition it is done for that. We say blood can be collected to detect or to identify bacterial composition. That is reason number one. Bacterial contamination, that is number one. We also have to understand or identify viruses. That is number two. Antibodies in the blood serology. Again, the antibodies which we said we need to understand that is number three. The antibodies, remember that these are the cells in the body that prevents or that fight against infections. And then we have got urine provides for detection of bacteria again, which we also identify that there is bacterial load which must be analyzed in urine. Cast red or white blood cells composition which shows infection in the urine again. Semen is also collected as a sample, as a biomedical sample to detect in fetal conditions and check positive vasectomy status. If you can check, if you can understand, uh, people or many some in most cases in regard to the semen there, we are saying, or even any other people which also we say women can also produce in terms of the lubrication that they release there. What happens is that it is also collected to identify the possibilities of individuals to have children. It is contained or it can be verified through semen examination. Semen examination can be done to identify possibilities of having children and also check positive vasectomy status. Vasectomy as to say that the cutting of tubes and also to prevent it is a contraceptive measure which can be done to see that the contraception that was done, the cutting and tying of tubes was successful. So the semen it can be collected to identify and evaluate that there is no sperm cells or egg cells that can be found within the semen there. And then feces can also be done to detect bacteria, viruses, and parasites, which we should understand quite often that feces examination or collection can be done to identify bacteria load or also viruses and other parasites, which can be gastric parasites or gastric uh, bacteria that can be found in the stomach. So they can also be passed in the urine there, I mean in the feces. Hence, they can be taken for examination to check in regard to that. And then aspirates provides evidence for bacterial infection again. Sputum is collected if pneumonia or respiratory tract infection is suspected. I'm sure that you can agree with me that the sputum test, it is also taken to identify extreme coldness of the body as well as respiratory tract infections that are also suspected. So such ideas or such samples are collected for purposes such as those. Uh, Let us quickly look at this one, specific gravity, which is also our our last aspect. Specific gravity. Specific gravity, basically, it is a measurement of one liquid in regards or in comparison to another. I can give it as uh, a measurement of one liquid in relation to another in regard to its, we can say, mass. So what happens there is that specific gravity is actually a measure of density compared in this regard. It is a measure of density compared to water. It depends on the quantity and density of solids, which we are saying in regard to this, it can be a comparison of urine to what to water, urine uh, versus water. We are saying when these two are mixed together, in regard to specific gravity, it measures the weight or the density, it can measure the weight or the density of one of the liquid to in comparative to another. So what we need to understand is that specific gravity or the quantities of solids that are found in each of the different liquids that are given defines the mass or defines the density. So the one which is heavier, you would find it going down than the other, which we can say if I can give uh, a container such as to say that is our container there, that is our container. So we can say if these two are mixed together, you would find out that at this level, we would find out that urine is found at the base. Urine is found is at the base of the container, is at the base, and then you find out that there is water, as to say water is on top. So then when water is on top, what it means is that the specific gravity or the gravity or the density of water, of urine is heavier compared to water. So what I'm meaning to say here is that specific gravity, it actually measures the densities of two different liquids when mixed together. What is their densities in relation to each other? So as you can look here, we have got the first part of it is water, and then the underneath part, it is urine. So when mixed together, we always see a division that marks the amount of water which is on top, and also the lower part which indicates the amount of urine which is also underneath. And then normal urine has a specific gravity which changes from 1010 to 1025. That's the specific gravity of urine, which is normal. And then specific gravity is determined by urinometer. To say the urinometer is actually used to measure, as to say it's an instrument that is used to measure specific gravity. It is the instrument that is used to measure specific gravity, urinometer. And then a quantity of urine is placed in the cylinder, and urinometer is placed in the urine and is given a slight spin with the thumb and the second finger so that it does not become attached to the sides of the cylinder. That's basically that's actually the apparatus that is used in regard to that. And then chemical analysis, if you can check to it, that we are saying it is also done to determine the contents of proteins, of blood, of ketone and glucose and all other gall pigmentation in the urine. There are test strips and also tablets available. For example, the test tape, which can also be done to identify chemical analysis in the urine. So chemical analysis is actually used, it is actually done to identify the solids or the amount of soluble substances that has been found in the that are found in the urine, which we are saying these are usually defined as, they say, they are residues. They are the residues, these ones. They are the residues that are, to say, they are the leftovers which are disposed as what? As waste. So please, class, take note that this is the first chapter of our medical N5, and I wish that you are going to enjoy and get the best. Thank you very much, and look forward to the next topic. Please don't forget to subscribe and turn on your notification button. Metric help, share to a friend and another friend. Thank you so much, and have a good day.